My name is Amanda. I am going to be going over protein synthesis. I have a word bank on the side so that I don't forget any of the process and to make sure that I can go over anything as thoroughly as possible. Protein synthesis also consists of two different processes. Those go by transcription and translation. I wrote that really small. That I'll have to do. Okay. So we're going to begin with basic cell structure. This is going to be in eukaryotic cells. So the surrounding area is going to be cytoplasm. And we also have our nucleus. From here, we also have our DNA. Okay, so this is our DNA. DNA is also known as a template. With our DNA, we also have coding regions. And we have non-coding regions. These are also known as our introns and extrons. So I will go ahead and label that real quick. So our blank spaces right here, these would be our introns. I'm going to label it with an I, just so you guys get a little bit of an idea. And this highlighted part would be our extrons. Okay. So along with our introns and extrons, we also have start signals and stop signals. They are also known as promoters and terminators. So that would be our stop signal. This is our start signal. But again, our stop and start signals aren't just stop and start signals. They are also called promoter and terminator. So in these, in our DNA, we also have something called triplets. That's something I want you to keep in mind. Our promoter is the start signal of transcription. So we're going to put that on the side.
together as part of the DNA sequence.
So another thing is the information in DNA is being copied into RNA, correct? I was going to write it down, but if I said it, then it should be fine. I think you can understand. So from there, so this right here is RNA. The process of DNA turning into RNA is what transcription is. So we're going to also label transcription. This right here is RNA polymers or merase.
We also have these little exits.
These are our nuclear pores. Okay, so next is we have these little ends right here. These ends include the 5-guanine cap, along with the 3-poly tail. Correct? Okay, so mRNA processing involves multiple stages. There's removal of introns, splicing together extrons, the 5-inch guanine cap, and the poly A tail. And all of this happens in the nucleus. Okay, from here, this is then mRNA. The mRNA is going to exit through the nuclear pores, and it's going to move on to the next step. The next step being the ribosomes. We have the small ribosomal subunit. Okay. From here, the mRNA has traveled, and it is now binding to the small ribosomal subunit. And we have these codons that sit on it. So right here we have AUG. AUG are our start codons. Above the AUG. Besides from it being just there, we also have tRNA. And they are all surrounding. on the outside, but we have tRNA also right here. And tRNA has anticodons. tRNA all also carry amino acids. And there are different types of amino acids. And this would be methionine. So along with mRNA and translation in general, so it's sort of divided into two parts. This over here inside of the nucleus is all transcription, and this over here is translation. Translation also consists of different stages. Translation consists of initiation, elongation, and termination. So not only are the codons right there, there's also codons throughout the whole mRNA. And it's in threes. And it sort of just continues. Just like that. The large ribosomal unit can come together now. So our anticodons have complementary anticodons. So the complementary codons for AUG would be UAC. I wanted to include this image to give you a little bit of a better idea for the anticodons and the codons. This image came from some homework and I felt like it would help with the understanding of it all. We also have stop codons. They are the stage of termination. In this case, it would be UGA. And that is protein synthesis.